Okay, imagine this. You're standing with me right now in Namche Bazaar. We're way up, 3,440 meters. For most of us, the air would feel really thin, wouldn't it? Every breath of it. A bit of a struggle, heart pounding. Yet, you look around and you see Sherpa Highlanders moving through this demanding terrain with, well, almost an effortless grace. Now, this difference, it isn't just about training. It's a story written deep in our genes. We often hear about maybe a single Nepal gene, kind of a myth almost that explains this. But today we're going to unpack that idea a bit. This deep dive is all about revealing a kind of hidden toolkit. So not one switch, but, a whole collection of fascinating adaptations. Shaped by mountain, sure, but also by human movement, even the food people eat over generations.
That's right. And we'll be exploring a really fascinating altitude gene. It's got ancient origins, actually linked directly to Denisovans. Then we'll get into a unique story about blood physiology that might just, well, flip your expectations about high-altitude living. And for a bit of a twist, we'll uncover some surprising genetic echoes from Nepal's lowlands, the malaria-prone areas. It shows how different environments leave really distinct genetic marks. Our mission really is to understand Nepal's genetic landscape not as one single thing, but as this vibrant, multifaceted testament to human adaptation.
Exactly. So get ready for some, I think, surprising facts and maybe some aha moments that will shortcut you to being really well informed about this incredible part of the world.
Okay, let's start high up then, back in Namche Bazaar. For most people, low oxygen, it makes your lungs work harder, your heart pounds. It's a real physical challenge.
Yeah.
But for the Sherpa, this is, well, it's their natural environment, their home. So biologically, what's actually going on? What's their secret? Well, a key part of that secret is a pretty famous altitude gene. It's called EPAS1. And what's really fascinating here is that this specific version, this variant, it shows clear signs of ancient introgression, which means it was inherited from Denisovans, the archaic human group. This isn't just a theory. It's something shown again and again in genetic studies of Tibetans and Sherpa, who are, neighbors, living in Nepal's high-altitude regions.
Wow, Denisovans. So this ancient connection directly helps them today.
Yes, exactly. Selection, natural selection, pushed this specific EPAS1 variant to become really common because it genuinely helps the body handle low oxygen more efficiently. It confers a real advantage.
Okay, so it's a proven advantage. And you can actually measure this. I read about a study genotyping a lot of people across Eurasia.
That's right. A significant study genotyped 1,507 people, and importantly, 1,188 of those were from Bhutan and Nepal. And they found this Denisovan-like EPAS1 genetic signature, the haplotype. It literally climbs with altitude.
It climbs with altitude. What does that mean exactly?
It means there was a remarkably strong correlation. The air value was 0.75, which is, well, very high in this kind of research.
Yeah.
It basically means the higher up a village is located, the more often you find this adaptive haplotype in the people living there.
That's incredible. So it really makes you think, doesn't it? If altitude shapes genes that strongly, should we maybe think of the Himalayas as this, I don't know, continuously running laboratory, actively shaping human evolution generation after generation?
That's a great way to put it.
Sure.
Strongly suggests that, yes, the selective pressures are ongoing, constantly refining that genetic toolkit. It's evolution in action right before our eyes, essentially.
Okay, let's shift gears a bit and talk about blood, because
Right.
what happens to our blood at altitude is obviously crucial. Many of us, when we go high up, our bodies react by making more hemoglobin to carry more oxygen. But that has a downside, right? It thickens the blood, can be risky, sometimes called blood sludge. Sherpa physiology, though, seems to kind of bend these rules. How?
Yeah, it's really interesting. Several studies consistently show that Sherpa maintain a lower hemoglobin concentration compared to, say, Andean populations living at similar altitudes.
Ooh. But they perform just as well.
Exactly. Just as well, if not better in some cases. This less sludge, same oxygen strategy, as you call it, seems largely down to differences in their plasma volume and their total hemoglobin mass. It's a more, let's say, efficient way to manage oxygen delivery.
More efficient, less risky. Makes sense. And you mentioned it's linked to genes like EPS1.
Yes, and another one called EGLN1. These genes are involved in the body's response to hypoxia, or low oxygen. Selection seems to have favored variants in these genes that support this different physiological strategy.
And are there real-world health benefits observed from this, beyond just athletic performance?
Absolutely. Research actually shows that Sherpa women who naturally have these lower hemoglobin levels tend to have better reproductive outcomes at high altitude, fewer complications during pregnancy, healthier babies.
Wow. That's a powerful natural advantage, then. directly impacting survival and the ability to thrive there.
It really is. It's a critical part of their adaptation.
So this raises a key question for me, and maybe for you listening. Is this physiological edge mostly genetic, hardwired? Or is it something perhaps that one could train towards, develop over a lifetime with enough exposure?
That's that's the million-dollar question, isn't it? And the scientific literature suggests it's likely a mix. The underlying genetics, driven by that strong selection pressure on genes like EPAS1 and EGLN1, definitely provide a significant head start and inherited advantage. But environmental factors, acclimatization over a lifetime, developmental acclimatization from childhood, they also play a really crucial role. It's rarely just one or the other.
Nature and nurture interacting.
Precisely. A complex interplay, constantly shaped by that high-altitude environment.
Okay, so we've seen these incredible adaptations for thin air, but let's zoom out a bit. Where did this whole genetic toolkit come from? Because Nepal isn't some isolated genetic island, right? It's actually a major crossroads.
Absolutely not isolated. It's a vibrant crossroads, historically and genetically. If you look at the big picture, Nepalese populations are positioned right between Tibeto-Burman speaking groups coming from the north, like Tibetans, and Indo-Aryan speaking groups from the south, from the Indian subcontinent.
And you can see this mixing in their DNA.
Clearly. Genome-wide studies looking at, thousands of genetic markers across over 1,200 individuals from various Nepalese groups, they reveal definite genetic substructure. So, for example, the Sherpa, as we've discussed, are genetically very close to Tibetans. They appear quite distinct, maybe reflecting their specific history and adaptation. But many other groups in Nepal, like the Rai, the Magar, the Tamang, they carry quite sizable northern Tibeto-Burman-related ancestry. Then, in contrast, you have Indo-Aryan groups in Nepal showing much stronger genetic ties to the south. It tells us Nepal isn't one bloodline. It's this beautiful, complex braid of different ancestries woven together over time.
A braid of ancestries, I like that. And ancient DNA can actually put timestamps on this weaving process.
Yes, exactly. There was a fascinating project analyzing 38 ancient individuals from 7 different sites in Nepal. These remains dated from about 1500 BCE, way up to around 650 CE. And what they found was, on one hand, long-term genetic continuity in the highlands related to today's Tibetans, showing those high-altitude adaptations have deep roots. But crucially, they also found these smaller but definite threads of genetic input from elsewhere mixing in over time.
So the toolkit has ancient highland roots, but it's also been added to and modified by these later connections.
Precisely. It really solidifies this idea of Nepal's genetic landscape being diverse and having deep layered roots, which, brings us back to that question. If identity is really this braid of diverse ancestries and adaptations, should we maybe retire the phrase the Nepal gene and instead talk about Nepal's rich and varied genetic landscape? It just seems much more accurate, doesn't it?
It really does. A landscape, not a single point. That makes so much more sense. And to illustrate this further, sometimes you hear about the EDAR variant. That's the gene linked to things like thicker hair, certain tooth shapes, often seen as an East Asian marker. How does that fit into Nepal's story?
Right. EDAR is a good example. By looking at variants like EDAR and also things like maternal line haplogroups, which trace ancestry through the mother's side, researchers can effectively trace the paths of these different genetic inputs. So they can see how East Eurasian components, for instance, flowed into various Nepalese groups over time. It's a powerful reminder, really. A single visible trait or a single gene variant can travel with migrations, with marriages across mountains. It tells part of the story, but it never truly defines an entire people or their complex history.
Fascinating. Okay, let's travel now, metaphorically, down from the mountains to a completely different part of Nepal, the Terai region. This area is low-lying, borders India, and it's known as Nepal's malaria belt. So a very different environment, very different pressures.
Exactly. Here, the big evolutionary story isn't about oxygen, it's about pathogens, specifically malaria. The selective pressure from malaria over centuries has really shaped the red blood cell genetics of the people living there.
How so? What kind of genetic signature does malaria leave?
One of the key examples is G6PD deficiency. It's an enzyme deficiency affecting red blood cells. A national survey looked at over 4,000 people across 12 malaria endemic districts in the Terai. They found G6PD deficiency in about 3.5% of the overall population sampled. It was slightly higher in males, 4.1%, than females, 2.1%, which is expected due to how it's inherited.
Okay, 3.5% overall. But I remember reading there were big differences between groups.
Yes, and this is where it gets really interesting, showing how localized selection can be. Some specific subgroups within the Terai had much, much higher rates. For instance, in the Mahato subgroup, the The prevalence was a staggering 17.6%.
Wow, nearly 18%. Why would a deficiency like that become so common?
It's believed to offer some protection against malaria, particularly severe forms. So where malaria is intense, individuals carrying the G6PD deficiency gene variant had a survival advantage, and the gene became more common over generations. It's a classic example of balancing selection, where a gene variant is beneficial in one context, fighting malaria, even if it might have downsides in another.
And that downside is important today, right? Especially for treating malaria.
Hugely important. This isn't just historical genetics. It has direct clinical relevance now. G6PD status directly impacts how safely someone can be treated with certain anti-malarial drugs, specifically primaquine. Primaquine is really important for the radical cure of Plasmodium vivax malaria, which is common in the Terai. But if you give it to someone with G6PD deficiency, it can cause severe hemolytic anemia, basically. Their red blood cells break down rapidly.
That's serious. So these genes, shaped by ancient mosquito maps, have real consequences for modern medicine there.
Absolutely. It underscores how deeply environmental challenges get etched into our genome and how that interacts with healthcare today.
Which leads to a pretty critical public health question then. Given the risks, should G6PD testing be routine in clinics in the Terai before giving drugs like primaquine?
Ideally, yes. From a purely medical safety standpoint, most public health experts would strongly advocate for routine testing before administering primaquine for the radical cure. The potential for severe adverse reactions is significant. However, we also have to be realistic. In many parts of the Terai, like in many resource-limited settings globally, there are very real constraints: logistics, cost, access to reliable testing.
Right. So the ideal medical practice bumps up against practical challenges on the ground.
Exactly. It's a difficult balance that health systems constantly navigate.
Okay, so bringing this all together now, we've been from the high Himalayas to the low-lying Terai. What's the big takeaway? It seems pretty clear there's no single monolithic Nepal gene. That idea just doesn't fit the evidence we've discussed.
No, not at all. It's far too simplistic. Instead, what we've uncovered today is this incredible Nepal genetic toolkit, plural. It's a powerful testament to human adaptation that's been fast, highly localized, and layered over time. It's been shaped by the extreme altitudes of the Himalayas, certainly, but also by the complex history of human movement across these landscapes, north to south, south to north, and by the specific health challenges of its diverse ecologies, like malaria in the plains.
So different tools for different jobs, essentially. EPS1 and its partners for efficient oxygen use high up.
Right, enabling that less sludge, same oxygen blood function without the usual risks.
While G6PD deficiency becomes more common in the plains as a defense, albeit a complicated one, against malaria.
Exactly. And woven through all of this, across the Middle Hills especially, you have centuries of movement braiding together those Indo-Aryan and Tibeto-Burman genetic threads. It creates this unique population structure, this intricate tapestry.
So the truly special thing isn't some idea of genetic purity or finding one magic gene. It's actually this extraordinary dynamic story of adaptation, of resilience, of diversity playing out over millennia.
That's precisely it. It's the complexity and the responsiveness of the human genome in the face of diverse challenges that's truly remarkable here.
It really makes you wonder, doesn't it? What more can we learn about human resilience, not just in Nepal, but everywhere, when we look beyond those simple labels, beyond single genes, and really embrace the rich, messy, fascinating complexity of our shared genetic past and present?
A perfect thought to end on. There's still so much to uncover about these intricate stories written in our DNA.